Hey, welcome to the video for 7.4. We are going to finish talking about the respiration pathway, specifically that oxidative phosphorylation step. So we're going to talk about what happens in both aerobic and anaerobic respiration, and so we're going to talk a little bit about lithotrophy and where that fits in. Okay, so section 7.4 is called respiration and lithotrophs. The objectives for this section is you need to be able to describe how NADH transfers electrons to electron transport proteins of the electron transport system, and ultimately to the terminal electron acceptor, such as oxygen in aerobic respiration. And you need to be able to explain how the transfer of electrons from one protein to the next yields energy and pumps H plus across the membrane. Okay, so before we really start into the details of this section, we need to talk about respiration differences between eukaryotic and prokaryotic cells. We've hit this a little bit as we've gone through this chapter, but let's discuss the difference. So as far as respiration, so glycolysis, the Krebs cycle, these are both the same between both prokaryotes and eukaryotes. Okay, where they differ is where the electron transport chain is. So with both prokaryotes and eukaryotes, glycolysis is going to happen in the cytoplasm. Okay, in prokaryotic cells, the TCA cycle is in the cytoplasm. So in the presence of a final electron acceptor in eukaryotic cells, mainly oxygen in eukaryotes, what's going to happen is that oxygen is going to trigger the movement of pyruvate and those electron carriers from the cytoplasm into the mitochondria, where it's going to then enter the Krebs cycle and then go to the electron transport chain for oxidative phosphorylation. Now, the biggest difference between these two things is in prokaryotes, that electron transport chain is embedded in the cell membrane of the cell. Okay, so where they occur are different between the two types of cells. So here in a prokaryotic cell, we're going to have all these enzymes that are embedded in the membrane that are going to remove the electrons from their electron carriers, and we're going to use those electrons to pump these hydrogen ions out of the cell to make that proton motive force, that hydrogen concentration gradient. Okay, which then can come around and be used in ATP synthase and causes it to turn and it makes ATP. Now in mitochondria, we have what's called a double membrane. So we have an inner membrane and an outer membrane. Okay, so the electron transport chain is embedded in that inner membrane of mitochondria. So when it uses its redox enzymes to remove the electrons from the electron carriers, it's going to move those electrons through the different enzymes, and it's going to use those electrons to power the movement of those H plus ions into the intermembrane space, so the space between the outer and inner membranes. So that's going to create that proton motive force in the eukaryotic cells, which then can be used to go through ATP synthase, which is embedded in that inner membrane to make ATP. So the processes are really similar. We have similar energy production. Glycolysis is going to give us 2 ATP in both scenarios. Krebs is going to give us 2 ATP in both. Where we're going to differ is how much ATP is made at the end in this electron transport chain. Okay, so same but a little bit different. Okay, so we are back to our overview slide. We talked about glycolysis and the fact that we made a little bit of ATP, but mostly electron carriers. We have the oxidation of pyruvate, so we convert pyruvate to acetyl-CoA. We make some carbon dioxide. We make some more electron carriers. Pyruvate goes into the TCA cycle for respiration. We're going to make a little bit more ATP. We're going to make a bit more carbon dioxide. We're going to make a lot of electron carriers. Okay, so at this step, we're now going to go to oxidative phosphorylation after the TCA cycle. Okay, so we need to look at what's happening with these electron carriers that we made through this process so far and what happens there. Okay, so let's talk about what happens here in this respiration portion of this chain. Okay, so we have gone through glycolysis and TCA. We have removed all the electrons possible from the substrates we started with. So whether it was carbohydrates, proteins, lipids, acidic acid or sugar acids. No matter what we've gone through, we have now removed as many electrons as possible. Okay, we've added them to our electron carriers. So those electron carriers are going to travel to the membrane where it's going to interact with a bunch of redox enzymes that are embedded in that membrane, which we refer to as either the ETS, electron transport system, or ETC, or the electron transport chain. Now I'm going to use ETC. Our book uses ETS. They're both the same thing. Okay, so the important thing about this oxidative phosphorylation step, this electron transport chain step, is that we're going to have a flow of electrons that we have now recovered from the electron carriers, and it's going to flow through these enzymes in the membrane. And while it's flowing through these enzymes in the membrane, it's going to power these proton pumps, which are going to pump the hydrogen ion, that H plus ion, outside of the membrane, and it's going to generate that H plus concentration gradient, which we refer to as the proton motive force. Okay, so this is going to allow for a change in concentration gradient, so those H plus ions are going to start to diffuse back in to the membrane through that ATP synthase. Now, one of the important things besides making this proton motive force that the electron transport chain does is it's going to have a oxidizer or an oxidizing agent. So something that can remove the electrons from the electron carriers and move them through the transport chain. Okay, so in aerobic respiration, our oxidizing agent is oxygen. Makes sense, right? Oxidizing agent, oxygen. Okay, so this oxygen, this final electron acceptor, is going to be what pulls these electrons through this chain in that membrane. Okay, so for anaerobic respiration, we're going to have a final electron acceptor as well. It's just not going to be oxygen. And the thing is, oxygen is the best oxidizer, so the strongest electron acceptor that we have for this process. So in anaerobic respiration, the final electron acceptor in those processes, whether it's sulfur, iron, nitrogen, uranium in some cases, that is not going to be as strong and be as efficient as that oxygen. So we're not going to make as much energy because our electron acceptor is not as strong as oxygen. Okay, so at the end of the electron transport chain, we have finally finished catabolism. Okay, so for glucose, we have finally harvested and used all the energy that we could from that glucose molecule. Okay, so here we are. We're back here. So we have gone through the TCA cycle. What we are caring about are these electron carriers and the electrons they can't carry and their protons. So all of these electron carriers, so the 4 electrons and the 2 NADHs here, the 2 NADHs here, and the 6 NADHs and 2 FADH2s here, are all going to travel to the electron transport chain, where they can participate in oxidative phosphorylation. Okay, so at the end of the TCA cycle, we have all these high energy electrons. Now what happens? Okay, so oxidative phosphorylation is the process where we're going to convert ADP to ATP by adding an inorganic phosphate to the ADP molecule by a process called phosphorylation. So the enzyme that does this phosphorylation just means we're adding a phosphate group, is going to be that ATP synthase molecule that we keep talking about. So this is what's going to take us from our low energy ADP molecule to our high energy ATP molecule. Okay, so this is what happens. This is the whole point of this electron transport chain, is to use these high energy electrons to make ATP. Okay, so let's talk about where we are. So we have now shipped all of our electron carriers, all those high energy electrons that we got from glycolysis and TCA. So it is now at the membrane ready to go into the electron transport chain. Okay, so this is what shows us what the electron transport chain kind of looks like. So it's a series of 4 different enzymes. Okay, and the whole purpose of this electron transport chain is to oxidize NADH to pull the electrons from it and the hydrogen ions. So the electrons are going to go into the enzymes. Okay, and what's going to happen is it's going to power those proton pumps, which are going to take the hydrogens that also hitched a ride in on the electron carrier, and it's going to pump it out of the membrane to start to make that H plus gradient to make that proton motive force. Okay, so the electrons are going to be pulled through this whole chain, and it's going to pump hydrogen outside as we're going the whole way. Okay, so we're going to, at this step, we're going to use that oxygen in our cellular respiration. This is aerobic. We're going to take oxygen and we're going to split it, and it's going to make water. Okay, so this is where we get the water in the respiration pathway. Okay, so that final electron acceptor is going to be what pulls these electrons through this chain of enzymes as we go through, and it's going to power these proton pumps. So in aerobic respiration, oxygen is the final electron acceptor, so it's going to be the one that pulls these electrons through. It's going to be the most efficient way to do this process. Okay, so at the end of the electron transport chain, we have this proton motive force. We have this H plus gradient. Okay, so what's going to happen is that H plus gradient, we're going to now have a high concentration outside of the cells. What's going to happen as it goes through, it's going to find an ATP synthase, and it's going to travel through the ATP synthase. And now while this happens, it's going to turn this little ratchet, this little motor here, and it's going to attach the phosphate to the ADP molecule to make ATP again. Okay, so this is kind of what it looks like. This is the part that turns. We have our little ratchet. We have our channel where the H plus comes through. Okay, one of the really interesting things is that one of the differences between our electron transport chain and that in bacteria and archaea is that the ATP synthase is different. So bacterial ATP synthase, while it does the same job, it is a little bit different structurally than our ATP synthase in our mitochondria. So this enzyme, this ATP synthase, is actually a really important drug target. Okay, because it's only found in bacterial cells, that means it's only going to affect the ATP synthase for bacteria and not our ATP synthases. Okay, so there's different tuberculosis and methicillin-resistant Staphylococcus aureus treatments that specifically target this ATP synthase, so it shuts down energy production, which then shuts the cell down. Okay, so we have harvested all the energy from glucose. We have made our electron carriers. The electron carriers have made it to their final destination. They have given up their high energy electrons. They have now been oxidized so they can go back to their beginning parts in glycolysis or in the TCA cycle to be reused. Okay, so with oxidative phosphorylation, we have now made ATP. Now, how much ATP we make depends on the type of respiration we go through. So for aerobic respiration, one molecule of glucose is going to yield throughout the whole pathway, so glycolysis and TCA and oxidative phosphorylation, somewhere between 36 and 38 ATP. Under ideal conditions, it can be a little bit less than this as well. Okay, this is what happens in our cells. Okay, we are very efficient at making ATP. Our mitochondria make it so this happens that way. Now, bacteria, they rarely make as much ATP as our mitochondria make for our cells. Okay, they're much less efficient. Okay, and there's a reason for that, and we're going to talk about that in the next slide. Now, just a reminder, if we go through fermentation like a lot of anaerobic microbes do, we only make that 2 ATP from the glycolysis. We don't make any more energy than that. Okay, so that is the least efficient process in this whole energy making scheme. Okay, so it goes fermentation the least, anaerobic, and then aerobic respiration, which is the most efficient. Okay, so for aerobic, we can make around 38. Bacterial cells, we're going to make a different amount. Okay, so one of the problems with bacteria cells is where that proton motive force is being formed. So with bacterial cells, we are pumping the hydrogen ions out into the extracellular matrix, so outside environment. There's no way to trap them, which means there's no way to direct them to just using ATP like the mitochondria do for our cells. So that means this proton motive force is available to power a bunch of different things. So one of the things that this proton motive force can do is it can power the rotation of the flagella. So it will allow it to do the rotation and turn the way it should to power the bacteria to find different locations. Okay, so another way it could use H plus is that it can power different pumps in the membrane. Okay, so we have some antiports, we have some symports that can use it. One of the important ones is hydrogen. The hydrogen ions, the proton motive force, can be used to pump antibiotics out through these drug efflux pumps. Okay, so H plus comes back in through diffusion, and the antibiotic gets pumped out because that H plus powered that. Okay, because there's so many different areas where the proton motive force can be used, we take away some of that energy, some of that power that we've put into this proton motive force. So not all of that is going to make ATP. We still get more than we do in fermentation, but not a lot more because we're powering all these other different reactions. So this is the reason why bacteria are less efficient than mitochondria. Now I would know this and be able to repeat this and tell me about this on the exam. This is a question. Okay, so that's going to bring us to the end of respiration, and it's going to bring us to what happens if oxygen is not our final electron receptor. Okay, so remember, not all respiration is aerobic. We have anaerobic respiration. So anaerobic is going to yield less than aerobic respiration. Okay, because bacteria, unlike our cells, can use alternative terminal electron receptors. So whenever you see this term terminal electron receptor, it's just the same as final electron receptor. Okay, so we're going to use these different electron receptors to complete respiration, and they're just not as efficient as oxygen. So we have Geobacter, let me see if I can say this today, guys. Metalloreducens, which is a bacteria that we can find in the soil. They like to remediate pollutants, so they remove pollutants from the atmosphere and from soil. Okay, they are going to transfer electrons through catabolism to a bunch of different metals as their final electron receptor. So most of the time it uses iron as its final electron receptor. It's not as strong as oxygen, but it still does a pretty good job. But they can use uranium and other metals to do this job as well. We have Pseudomonas aeruginosa, which we know from one of the previous lectures is the bacteria that causes the colonies inside the lungs of cystic fibrosis patients. So Pseudomonas aeruginosa is going to use nitrogen as its final electron acceptor. Okay, we have a bunch that are going to use sulfur and sulfates for electron receptors. Pyrococcus furiosus. This is one of our archaea that are found down by the hydrothermal vents. So they grow at those really high temperatures. It's going to use elemental sulfur. That's what S0 means, is elemental sulfur. And it's going to use it as the electron acceptor, and this is going to make that hydrogen sulfide that is made down in the hydrothermal vents. That's then going to be used for lithotrophy by a bunch of different other bacterial species. Okay, so Salmonella enterica, this is one of our pathogens that affects our intestines. They're going to use sulfur as well. Okay, so all of these final electron receptors are going to pull the electrons through that electron transport chain, but they're not as strong as oxygen, so they're going to be less efficient than oxygen, which means we're going to lose energy because they're less efficient. So I would know some of these for the exam, so be able to tell me like iron, nitrogen, sulfur, or some of our alternative final electron receptors for anaerobic respiration. Okay, so that is 7.4. We have made it. Okay, we have now talked about the whole respiration pathway from glycolysis to the TCA cycle to the oxidative phosphorylation, the electron transport chain. We have gone through all of glucose catabolism from start to finish. We've talked about anaerobic versus aerobic, and we've talked about fermentation. Now, in the next chapter, we're going to start talking about photosynthesis, so what the photoautotrophs do. Okay, so we'll see you for the video for chapter 7.5 next.